Applications of cell and tissue culture. Clonal propagation in nature. The methods of plant propagation may be either asexual, by multiplication of vegetative parts, or sexual through generation of seeds. Multiplication generally or multiplication of genetically identical copies of a cultivar by asexual reproduction is called clonal propagation. The population derived from a single individual by asexual reproduction constitute a clone. Clonal propagation through tissue culture, popularly known as micropropagation. Use of tissue culture for micropropagation was initiated by G. Morrell in 1960, who found this as the only commercially viable approach for orchid propagation. Production of virus-free plants. Plants can be infected by a wide variety of microbes, viroids, viruses, mycoplasmas, bacteria, fungi, and nematodes. The infections greatly affect yield and quality of crop and ornamental plants. In some cases, infection can be eliminated by micropropagation of unaffected parts of the plant. The basic method of obtaining virus-free plants is culture of apical meristems. If the growing tip is cultured, then there is possibility to obtain virus-free plants which can be further multiplied. There are other methods like high temperature treatment, incubation of explants on suitable culture media containing malachite green or thiorizal, which can reduce virus infection and replication. These are, related to plant, not animals. So I'm gonna skip because I'm focusing on animals. So that's it. The ability to study animal cells depends largely on how readily they can be grown and manipulated in the laboratory. Although the process is more difficult than the culture of bacteria or yeasts, a wide variety of animal cells can be grown and manipulated in culture. Animal cell cultures are initiated by the dispersion of a piece of tissue into a suspension of its component cells, which is then added to a culture dish containing nutrient media. The culture media required for the propagation of animal cells are much more complex than the minimal media sufficient to support the growth of bacteria and yeasts. In the beginning, media used for cell culture consist of undefined components such as plasma, serum, and embryo extracts. However, in 1955, Harry Egley described the first defined media that supported the growth of animal cells. In addition to salts and glucose, the media used for animal cell culture contain various amino acids and vitamins which the cells cannot make for themselves. The growth media for most animal cells in culture also includes serum. It serves as a source of polypeptide growth factors that are required to stimulate cell division. Serum is the biological fluid most commonly used to support cell growth. So why serum is used? Because serum serve as a source of polypeptide growth factors that are required to stimulate cell division. Serum contains a variety of factors that facilitate cell attachment and growth, including carrier proteins, attachment regulators, defense molecules, enzymes and their regulators, growth factors, and hormones. Commercially, sera are obtained from a number of animal species. Calf serum is widely used due to its availability and low cost. Fetal bovine serum or FBS provides the greatest growth potential because of the high level of proteins and other associated growth factors. Cultures prepared directly from the tissues of an organism, that is without cell proliferation in vitro, are called primary cultures. The cells in a primary culture usually grow until they cover the culture dish surface. In most cases, cells in primary culture can be removed from the culture dish and made to proliferate to a large number of secondary cultures. This process can be repeated many times, but most normal cells cannot be grown in culture indefinitely, a process called cell senescence. For example, normal human fibroblasts, for example, typically divide only 25 to 40 times in culture before they stop. In these cells, the limited proliferation capacity reflects a progressive shortening of the cell's telomerase and telomeres and the repetitive DNA sequence and associated proteins that cap the ends of each chromosome. A culture of cells with an indefinite lifespan is considered immortal. Such a culture is called a cell line. The various types of cell lines are recognized into two types: finite cell line and transformed cell line. A cell line that has been transformed by a virus or mutation and proliferate indefinitely in a culture is called a transformed cell line. Transformed cells are immortal. They have no finite lifespan. They have a reduced requirement for serum or growth factors for optimal growth. They are anchorage independent. Also, there is a lot of contact inhibition, lots of density, loss of contact inhibition, loss of density dependent inhibition of proliferation. A transformed cell line often has an abnormal chromosome number, aneuploid, and overproduces different proteins. A finite cell line is a line of cells that will undergo only a finite number of divisions in cell culture before the cell will no longer divide. Cells are anchorage dependent and show density dependent inhibition. Finite cell lines are often considered to be closer in characteristics to the original cells from which they are derived and don't tend to have as much aneuploidy. The ability of cultured cells to grow indefinitely or their tendency to be transformed varies depending on the animal species from which the cells originate. Some widely used cell lines are 3T3 fibroblast of mouse, BHK21 fibroblast Syrian hamster, MDCK epithelial cell of dog, HeLa cells, then epithelial cell of human, CHO ovary Chinese hamster, R1 embryonic stem cells of mouse, H1 H9 embryonic stem cells of human. Among the most promising cell culture to be developed from a plant medical point of view are the human embryonic system cell lines. These cells, harvested from the inner cell mass of the early embryo, can proliferate indefinitely while retaining the ability to give rise to any part of the body. Embryonic stem cell lines. Cell culture systems. Two basic culture systems are used for growing cells. These are based primarily on the ability of the cells to either grow attached to a glass or treated plastic substrate like monolayer culture system or floating free on the culture medium like suspension culture systems. What are the applications of animal cell culture? Cell culture has become one of the most important or major tools used in cell and molecular biology. Some of the important areas where cell culture is currently playing a major role and briefly described below, like model systems. Cell culture provide a good model system for studying basic cell biology and biochemistry, the interaction between disease causing agents and cells, the effect of drugs on cells, the process and triggers for aging, and nutritional studies. Then toxicity testing. Cultured cells are widely used alone or in conjugation with animal tests to study the effects of new drugs, cosmetics, and chemicals on survival and growth in a wide variety of cell types. Especially important are liver and kidney derived cell cultures. In cancer research, since both normal cells and cancer cells can be grown in culture, the basic difference between them can be closely studied. In addition, it is possible by the use of chemicals, viruses, and radiations to convert normal cultured cells to cancer causing cells. This mechanism that caused the change can be studied. Cultured cancer cells also serve as a cell system to determine suitable drugs and methods for selectively destroying types of cancer. In virology, one of the earliest and major uses of cell culture is the replication of viruses in cell cultures in place of animals for use in vaccine production. Cell cultures are also widely used in the clinical detection and isolation of viruses as well as basic research into how they grow and infect organisms. Then cell based manufacturing. While cultured cells can be used to produce many important products, three areas are generating the most interest. The first is the large scale production of viruses for use in vaccine production. These include vaccines for polio, rabies, chickenpox, hepatitis B, and measles. Second is the large scale production of cells that have been genetically engineered to produce proteins that have medicinal or commercial value. These include monoclonal antibodies, insulin, hormones, etc. Third is the use of cells as replacement tissues and organs. Artificial skin for use in treating burns and ulcers are the first commercially available product. However, testing is underway on artificial organs such as pancreas, liver, and kidney. Some of the important products that are produced from animal cell cultures: enzymes, asparginase, pepsin, hyaluronidase, renin, and trypsin. Hormones: luteinizing hormone, follicle stimulating hormone, erythropoietin. Vaccines: foot and mouth disease vaccine, vaccines for influenza, measles, and mumps. Monoclonal antibody, cytokines, IFNs, interleukin. Tissue plasminogen activator, TPA. What is a genetic counseling? Amniocentesis. Amniocentesis, a diagnostic technique that enables doctors to remove and culture fetal cells from pregnant women, has given doctors an important tool for the early diagnosis of fetal disorders. The cells can then be examined for abnormalities in their chromosomes and genes during karyotyping, chromosome painting, and other molecular techniques. Genetic engineering, the ability to transfect or reprogram cultured cells with new genetic material, DNA, and genes, has provided a major tool to major molecular biologists wishing to study the cellular effects of the expression of these genes. New proteins. These techniques can also be used to produce these new proteins in large quantity in cultured cells for further study. These techniques can also be used to produce these new proteins in large quantity in cultured cells for further study. Insect cells are widely used as miniature cell factories to express sustained quantities of proteins that they manufacture after being infected with genetically engineered bacillovirus. That is gene therapy. The ability to genetically engineer cells has also led to their use for gene therapy. Cells can be removed from a patient lacking a functional gene, and the missing or damaged gene can then be replaced. The cells can be grown for a while in culture and then replaced into the patient. An alternative approach is to place the missing gene into a viral vector and then infect the patient with the virus in the hope that the missing gene will then be expressed in the patient's cells. That also comes under gene therapy. Then drug screening and development. Cell-based assays have become increasingly important for the pharmaceutical industry, not just for cytotoxicity testing, but also for high throughput screening of compounds that may have potential use as drugs. Stem cell culture. Stem cells have the remarkable potential to develop into many different cell types in the body. Serving as a sort of repair system for the body, they can theoretically divide without limit to replenish other cells as long as the person or animal is still alive. When a stem cell divides, each new cell has the potential to either remain a stem cell or become another type of cell with a more specialized function, such as a muscle cell, a red blood cell, or a brain cell. What are the unique properties of all stem cells? Stem cells differ from other kinds of cells in the body. All stem cells, regardless of their source, have three general properties. They are capable of dividing and renewing themselves for long periods. They are unspecialized and they can give rise to specialized cell types. Adult stem cells. An adult stem cell or somatic stem cell is an undifferentiated cell found among differentiated cells in a tissue or organ, can renew itself, and can differentiate into yield the major specialized cell types of the tissue or organ. The primary role of adult stem cells in a living organism are to maintain and repair the tissues in which they are found. Adult stem cells have been identified in many organs and tissues. In the 1960s, researchers discovered that the bone marrow contains at least two kinds of stem cells. One population, called hematopoietic stem cells, forms all the types of blood cells in the body. The individual hematopoietic stem cell is multipotent because it can give rise to complete range of blood cell types, both myeloid and lymphoid, as well as new stem cells like itself. A second population, called bone marrow stromal cells, was discovered a few years later. The adult brain also contains stem cells that are able to generate the brain's three major cell types: astrocytes and oligodendrocytes, which are non-neuronal cells and neurons or nerve cells. And embryonic stem cells, as the name suggests, are derived from embryos. Specifically, wait a minute, so we mentioned different types of stem cells like bone marrow stem cells, hematopoietic stem cells, bone marrow stromal cells, astrocytes, and oligodendrocytes. Embryonic stem cells, as the name suggests, are derived from embryos. Specifically, embryonic stem cells are derived from embryos that develop from eggs that have been fertilized in vivo. They are not derived from eggs fertilized in a woman's body. In vitro fertilization, not in vivo. I'm sorry. The embryo from which human embryonic stem cells are derived from are typically 4 or 5 days old and are hollow microscopic ball of cells called blastocyst. How are embryonic stem cells grown in a laboratory? Growing cells in the laboratory is known as cell culture. Human embryonic stem cells are isolated by transferring the inner cell mass into a plastic laboratory culture dish that contains a nutrient broth known as a culture medium. The cells divide and spread over the surface of the dish. The inner surface of the culture dish is typically coated with mouse embryonic skin cells that have been treated so they will not divide. This coating layer of cells is called a feeder layer. Cells used in co-culture to maintain pluripotent stem cells. For human embryonic stem cell culture, typical feeder layers include mouse embryonic fibroblasts, MEFs, or human embryonic fibroblasts that have been treated to prevent them from dividing. The reason for having a mouse cell in the bottom of the culture dish is to give the inner cell mass cells a sticky surface to which they can attach. Also, the feeder cells release nutrients into the culture medium. Recently, scientists have begun to devise ways of growing embryonic stem cells without the mouse feeder cells. This is a significant scientific advancement because of the risk that viruses of other macromolecules in the mouse cells may be transmitted to the human cells. Over the course of several days, the cells of the inner cell mass proliferate and begin to crowd the culture dish. When this occurs, they are removed gently and plated into a fresh culture dish. The process of replating the cells is repeated many times and for many months, and this is called subculturing. Each cycle of subculturing of cells is referred to as a passage. After 6 months or more, the original 30 cells of inner cell mass yield millions of embryonic stem cells. Embryonic stem cells that have proliferated in cell culture for 6 or more months without differentiating are pluripotent and appear genetically normal are referred to as an embryonic stem cell line. Once cell lines are established or even before that stage, batches of them can be frozen and shipped to other laboratories for further culture and experimentation. Then DNA isolation and purification. DNA isolation and purification involve disruption and lysis of the starting material followed by the removal of proteins and other contaminants and finally recovery of the DNA. The cells are lysed using a detergent and mixed with 1:1 mixture of phenol and chloroform. These organic solvents precipitate proteins but leave the nucleic acids DNA and RNA in aqueous solution. Before the phenol extraction, the cell extract is treated with protease like protease K for complete removal of proteins. Aqueous solution of nucleic acids can then be removed. Cell extract is there, DNA, RNA, protein, cell debris and mix with phenol, centrifugation, aqueous layer, DNA, RNA, interface, coagulated proteins, phenol. DNA can be purified by centrifugation through a CSCI density gradient, calcium chloride. Suspended DNA is mixed with cesium chloride and ethidium bromide and centrifuged for several hours. The DNA band is collected from the centrifuge tube, extracted with isopropanol to remove the ethidium bromide, and then precipitated in ethanol to recover the DNA. Plasmid purification. For plasmid purification, the bacterial cell extract is deprotonized, the DNA removed, and the DNA concentrated by ethanol precipitation. Ethanol precipitation involves the precipitation of nucleic acid by using absolute ethanol in the presence of salt and at a temperature of 20 degrees Celsius or less. In a plasmid purification, it is always necessary to separate the plasmid DNA from the large amount of bacterial chromosomal DNA. Separation of plasmid DNA from chromosomal DNA are based on several physical differences like conformation, size between plasmid DNA and chromosomal DNA. Plasmids and bacterial DNA differ in conformation. Plasmids and the bacterial chromosomes are circular, but during proper preparation of a cell extract, the chromosome is always broken to given linear fragments. Plasmids are covalently close to circular DNA and present in supercoiled conformation. If one of the polynucleotide strands is broken, the double helix reverts to its normal relaxed state. Then supercoiled molecules can be easily separated from non-supercoiled DNA. One method of separation is through alkaline denaturation. If sodium hydroxide is added to a cell extract containing linear and supercoiled DNA, then the hydrogen bonding in linear non-supercoiled DNA molecule is broken, causing the double helix to unwind and the two polynucleotide chains to separate. If acid is then added, these denatured bacterial DNA strands re-aggregate into a tangled mass. This insoluble network can be pelleted by centrifugation, leaving pure plasmid DNA in a supernatant. And DNA isolation and purification. Another method of separation is through cesium chloride buoyant density gradient centrifugation in the presence of DNA intercalating by ETBR. ETBR intercalation causes local unwinding of DNA helix, which reduces the molecular density of DNA. Large linear genomic DNA non-supercoiled molecules can bind more ETBR than supercoiled DNA owing to the difference in their topology. So low, high CSCL density genomic DNA, nicked circular DNA, supercoiled DNA. Because supercoiled plasmid DNA cannot be easily unwound, much less ETBR can intercalate. In contrast, linear DNA molecules are not as topologically constrained and can therefore absorb more ETBR molecules, resulting in an overall decrease in molecular density. And mini-prep method for isolation of plasmid DNA. Mini-prep method is a rapid, small-scale isolation of plasmid DNA from bacteria. It involves two basic steps: lysis of the bacteria and purification of the plasmid DNA. It involves alkaline lysis of cells to isolate plasmid DNA. Bacteria containing the plasmid of interest is first grown, then lysed with a strong alkaline buffer containing a detergent, sodium dodecyl sulfate, and a strong base, sodium hydroxide. The detergent breaks the membrane's phospholipid bilayer and the alkali denatures proteins involved in maintaining the structure of the cell's membrane. Through a series of steps involving agitation, precipitation, centrifugation, and the removal of supernatant, cellular debris is removed and the plasmid is isolated and purified.